Okay, class. Now we are on chapter 8. For the next, after we in chapter 7 for fertilization. Now we want to see how's the development inside the mother's womb, all those mitosis, meiosis, and so on. So catch up with me. We will continue. Okay, on this chapter. So for chapter 8, there are 6 subtopics that we want to cover. For the first one, what are cleavage and implantation? Second, organogenesis, how's the organ are developed? The gestation and pregnancy in a woman, labor and mechanism of parturition, lactation, and hormones development and lactation. So there's 6 parts of it, of this chapter. So as a learning outcome, you should be able to define terms and stages process in conception and development, able to describe hormonal controls for conception and pregnancy, and you should be able to compare implantation types in animals, be able to identify and explain fetal development, and explain mechanism of labor, parturition, and lactation. So as an introduction, during pregnancy as well as the development, there are 4 main stages. First one is fertilization. Second one, embryonic. Third one, the fetal development. Embryonic development, fetal development, and the parturition. Means once it is ready to give birth. Ovaries will release the eggs cyclically from the Graafian follicles. Maksudnya, we do have the cycle for each month. The fertilized egg is transported to the uterus where it develops into an embryo. So means the fertilized eggs from the fallopian tube will be transported within the uterus itself. So at the uterus, it will be implanted there, and now it can develop into embryo. So we have covered about the topic of fertilization. And then at the end of gestation, infant is born through the vagina or surgically through the abdomen if it is cesarean delivery. So what is conception? Conception is when the sperms and egg are meeting together and fertilization occur. Sperm is one of the tiniest cells of the body. Conception occur in the outer third of the fallopian tube, and if it is fertilized, the formation of zygote, what we call as the fertilized egg. So there are all of these are occurring inside a woman, not inside in man. So we can see for the ovulation, it a cyclic means every month for 15 days before next periods in fallopian tube for 12 to 24 hours. For the fertilization, during 12 to 24 hours since ovulation, and then the union of the sperm and eggs occur. This one are considered as conception. Implantation, if there's attachment of the fertilized eggs, or what we call as blastocyst, to the uterine wall. This took about 6 to 11 days after fertilization, and they are occurring at the uterus lining up to 2 days or 2 days or 48 hours after. So the conception requirement should be healthy egg and healthy sperm. Okay, both of them should be healthy. Second one, the place itself. The fallopian tube must be unobstructed, means not being obstructed with anything, and should be functional to receive the eggs from the ovary and to allow it to meet with the sperm. Then requirement next for the sperm ability to penetrate and fertilize the egg itself, and the requirement also the implantation means the the what we call as the attachment, the penetration, implantation, attachment of the embryo to the uterus, and healthy pregnancy is one of the requirement for the conception. Okay. Which contribution do they give? Okay, we do have contribution from the mother and contribution from the father. Contribution from the mother is the mRNA or genetics of the cytoplasm that carries instruction for early embryo development. They are haploid in the female genome and do contain centriole. And the contribution of sperms, they are also haploid male genome. They are containing centriole and they are activating this egg. So there are a lot of contribution that lead to the fertilization of a zygote. Then they are further developing into embryo. So there are a lot of the conception factors that give the chances of conceiving. For the age under 30 has higher chances of conception. Time of intercourse, higher chance it occur during the few days before or on the day of ovulation. So means you should know on the day of ovulation, the higher the capacity or the chances to get pregnant. Then the sperm's viability, the sperms live inside the woman's body for up to 5 days, and the egg's viability, if the egg is capable of being fertilized for about 12 to 24 hours after ovulation. So now we go to embryo development. Okay, development of embryo is the study of the progression of the cells from 2, 4, 8, 16, and become a complete set of a human. So this is what we call as embryology. Embryo is a subfield focused on early stages of early development. The zygote undergoes a series of mitotic cells division. So these are what we call as a cleavage. Okay, the cleavage. Cleavage is the division of cells by mitosis without any significant growth. Okay. So after fertilization, embryo will develop through 3 processes. Okay, first one, cleavage, what we call this one. Second one, gastrulation. And then third one is organogenesis. Gastrulation means there's gastrulate here. So what we call as gastrulation. And then we do have organogenesis. All of these layers become organ. So it is very, very important events regulate the development occur during fertilization and 3 successive stages that build the animal body. So now we go to the first stage of cleavage stages. Okay, this one is the picture under the microscope. From the 1, and it's become into 4, into 8, and then the name. There are names there. Okay, for the first one, it's called as zygote. Then second one, 4 cell stage. Third one is called as morula and blastula. Okay, we go further at the cleavage stage. It is the most period of rapid cell growth. cell division. This cell division is dividing, but without any growth there. So means the size is the same. The size will be unless the same. So from one cells, it's become two cells in only 6 days. It become into blastula. Zygote from one single cells into 2 cell stage to 4 cell stage, 8 cell stage, and we call as morula and turn into blastula. So zygote can occur in only few hours before the first cleavage division by this fertilization envelope. So this one what we call as the fertilization envelope. The nucleus is visible verily in the center. The second, for cell stage, remnants of the mitotic spindle can be seen. Okay, you can see the mitotic spindle there that have been just complete the second cleavage division. Then after that, morula, further cleavage division, is forming a multicellular ball that is only surrounded by the fertilization envelope. And then the blastocele cavity has begun to form. So this is what we call as the blastocele cavity. The blastula is a single layer of cells that surrounds a large blastocele cavity. So this is what we call as blastula. Although it is not visible here, the fertilization envelope is still present, means it's not as such as the first part here. And it will soon hatch from it and begin to swim to the uterus. Then we do have the second development, what we call as the embryo. It is starting from 9th week, the start of cleavage occurs. The embryo first undergoes division without any growth. The embryos enter the uterus at 16 cell state. Okay, and the embryo floats freely in the uterus temporarily. And what will the embryo gain for nourishment? Okay, the nourishment is from uterine secretion itself. Okay, blastocyst. We do have blastocyst cavity there. It's a ball-like circle of cells. Okay, this one is all of the cells like a ball. It begins when there's a lot of cells, 100 numbers of cell stage. And here, they secrete HCG for what? To continue producing hormones of progesterone. This one for the development of the uterus, uterine site. And then blastocyst implant in the wall of uterus by day of 14. Means almost 2 weeks there, almost 2 weeks of gestation. So the blastocyst start to implant or attach to the wall of uterus. Implantation. The HCG will stimulate the corpus luteum to continue to secrete progesterone by the uterus itself. The uterine lining continues to thicken, strengthen, then embryo can implant there. After 3 months, the corpus luteum degrades, and from the corpus luteum, the role are being taken by the placenta for nourishment for the embryo. And the uterine of the tissues begin to form placenta. Placenta itself can secrete estrogen and progesterone, not the corpus luteum. Then during the implantation, the blastocyst will be implanted 7 days after fertilization. They are what we call as trophoblast. Okay, this layer, the outer layer of the blastocyst, what we call as trophoblast. Okay, it's the epithelium of blastocyst that provides support, but it does not contribute to the embryo. For what? This trophoblast can initiate the implantation, the attachment to the uterus, and it will secrete enzyme that breaks down the endometrium lining so that it can attach there. And then it can expand region through cell division, and inside the cell of mass or embryoblast there, it can form like this, what we call as epiblast and hypoblast, lower layer as well as upper layer. Okay, once it reach the uterus, the trophoblast there and blastocele, we do have endometrium lining. Blastocyst will implant 7 days after fertilization inside the mother. Okay, this one is mother, this one is the baby. So the trophoblast will expand the region, okay, try to going and attach more on mother's blood vessels and cell walls of uterine. And then now the blastocyst develop into epiblast, the ones at the outer layer, and hypoblast. This one. Okay. Then the sequence of implantation, we do have extra embryonic membranes start to form in 10 to 11 days. Gastrulation will begin at 13 days. Trophoblast here can continue to expand its region by mitosis. They are trying to expand, and form finger-like projection into surrounding maternal tissue. Then the invasion can lead to erosion of capillaries, means the blood will be spilling out. And as implantation complete, gastrulation start to begins. What is gastrulation? Okay, we'll see after this what's the meaning of gastrulation. Okay, then the cells starting to move inward from the epiblast, from endoderm and mesoderm. Okay, you can see there are 2 layers of cells from endoderm and mesoderm. Extra embryonic membranes begin to form. You can see the extra embryonic membrane. Okay, starting from amniotic cavity, the capillary and so on. So from the chorion itself can be developed into, you see the color from the blue, green, and the red in color. It can be developed into ectoderm for the blue, mesoderm for the red, and the endoderm for the green in color. Then it's further developed into embryo with chorionic villi, and now it's try to connect to the maternal blood vessel, and placenta are now forming that replace the corpus luteum to get the nourishment. Okay. So now we call about the HCG, right? HCG that are secreted from the corpus luteum. So initially, the one who are secret HCG is what we call as this trophoblast. So the one who are secret is from this trophoblast. Okay, this trophoblast secret hormone HCG. For what the hormone? To prevent degeneration of the corpus luteum. So the corpus luteum will be there, stimulate the growth of corpus luteum. So from corpus luteum, it can increase the secretion of estrogen and progesterone, and to prevent any menstruation. If it's menstruation there, means it's higher chances for abortion will occur. Okay, if there's lack of HCG. So early in pregnancy, the level of HCG increases in the blood. That's why the color we can see HCG or urine are usually used for the detection of early pregnancy. So maternal recognition reflects various ways which the mother responds to presence of conceptus within her reproductive tract. Okay, so it is the process, some of the signal that prevents luteal regression, allow the corpus luteum to persist, continue to secrete progesterone. So means inside the blood of a mother, there's signal serum of progesterone will be higher or rising up at the constant level, going down for every single month. But during fertile mating, means during pregnancy, the maternal recognition, the corpus luteum will be there and not being demolished. So the level of progesterone will remain until the end of giving birth. And then if it is in cow and eve, the blastocyst will block the synthesis of uterine oxytocin receptor. Okay, why? Okay, receptor are blocked. Non-specific glycoprotein from white blood cell and trophoblastic cells also promote protein synthesis by the uterine glands. And this one is for the embryo survival. Okay, now we go to the next part. After blastocyst formulation, there's next part of gastrulation. Okay, this is morphogenetic process, means the morphology plus genetic. Okay, so they are arranging the blastula that only has three layers into 3-layer embryo. We do have ectoderm, mesoderm at the middle, and endoderm in the inner part. Mesoderm at the outer part, at the middle part, ectoderm at the outer part, and endoderm in the inner part. So these are gastrula, or what we call gastrula. Before this, at the primitive, we call as its gut or digestive tube. But they do have difference from animals to one another, from men, animals, and other, and the process is the same, but it is driven by the same general mechanism in all species. So gastrulation, we should know all those layers. From ectoderms, it can form exoskeleton. At the middle, or we call as mesoderm, it can develop into organ. And the endoderm, it can form inner lining of organs. So the organs do have lining, right? So the endoderm itself can form the inner lining. Ectoderm forms the outer layer of the gastrula. Mesoderm will fill the space between the endoderm and ectoderm. The endoderm will lines the embryonic digestive tract. Okay, so there's a lot of layer of gastrulation, 3 layers. Okay, 3 layers, endoderm, ectoderm, and mesoderm. But from 3 layers, they do have 4 extra embryonic membrane. They do have 4 membrane. Okay, so the trophoblast during this gastrulation, this trophoblast yang paling hujung ni, derived from epiblast and adjacent endometrial tissue. And from this tissue, they will become or develop into placenta. The four extraembryonic membrane is what we call first as amnion, second one chorion, third one yolk cell, and the fourth one allointois. Okay, 3 layers of membrane for 3-layered embryo with 4 extra embryonic membranes. Those are 4 membranes that are formed during gastrulation. Okay, so after implantation there, we can see the placenta now are formed, and then the chorionic villi projection of the blastocyst will develop, and the cooperation of the cells of the uterus to form the placenta. And now the embryo is surrounded by the amnion fluid. Okay, we do have chorionic. This forms of villi. Okay, villi projection. And then we do have amnion. Okay. Amnion is a fluid filled sac. Satu kantung lah. Okay, from this amnion itself, dia akan jadi kantung. Satu ni, amnion ni akan jadi kantung. Chorion akan jadi villi. This chorion ini akan jadi villi yang banyak. And umbilical cords form to attach embryo to the placenta. If there's defects in gastrulation, there are types of disease. Okay, what we call as meningohydroencephalopathy, development of abnormality for the central nervous system, and then cyanomelia, what we call as mermaid syndrome, where congenital deform, which the legs are fused together. So the legs, both of it, are fused together and giving them appearance of mermaid tails. What if there's ectopic pregnancy? Ectopic pregnancy are occur if the embryo implants in the fallopian tube, means not inside the uterus itself. So mostly the cell, the lateral wall will be ruptured, means the baby also cannot be safe. So it can both give a higher risk to both of the mother as well as the baby itself. Okay, I think that's all for today's part of development. Next, we will continue to the organogenesis and so on. So stay tuned with me. See you for the next part of this chapter. Thank you. Assalamualaikum warahmatullahi wabarakatuh.